All right, so today I'm presenting a framework for diffusion fiber-based analysis. Okay, yeah. So framework for diffusion fiber-based analysis of T1 T2 ratio map. This is done as a project of NIRAL. So the title is kind of not so obvious about what we're doing. So I really want to stress out the biological background of our motivation. What we're interested in doing is this process called myelinations in white matter. So our brain is separated into gray matter and white matter. And white matter is sort of like the network in our brain that connects one part to each other. Myelination is a process that happens within white matter, especially around fiber tracts that travel through white matter. So it happens as your brain develops. And it plays a very important role in human learning. For example, study shows that musicians who have been practicing pianos for years have high levels of myelination in certain part of their brain. So we want to create a tool to help researchers to evaluate how this process of myelination is related to several factors like gender, age, and especially mental disability. So then problem arises for how we could actually measure myelination, right? So common ways of doing this in neuroscience is you do a brain scan of the brain and look at the image to see if certain parts light up. And we normally use DTI to look at myelination as we could compute certain diffusion properties from the tensors. However, the problem with DTI is it's not only sensitive to myelination, but it's also sensitive to many other factors like fiber densities, like axonocytes. So a scholar called Glasser finds a better approach to estimate myelination, which is called T1w, T2w ratio. It's a computed image from T1 relaxation time image and T2 image. And the punchline is they find this type of image especially sensitive to myelin contents, while it's not sensitive to other factors. However, the raw T1 T2 ratio map is only qualitative, which means it doesn't make really much sense in comparison. So what we really want to do is a quantitative research of myelination, so which means it needs a process of intensity calibration, which I'm going to talk later. And another part of this project is why do we want to do it specific to fibers, right? Let me talk about another popular way of doing neuroimage analysis first, which is called region of interest analysis. The region of interest analysis pretty much segment the brain scan into several regions, usually based on lobe. So here I sort of mark out a part of the lobe that's in the brain. And as you could see here, that the problem of ROI in our case is that this lobe region contains lots of fiber tracts, which is denoted by different colors here. And also, more importantly, it does not include all parts of the fiber, but only parts of it. But since myelination is really a process that happens all along the fiber tract from one part to another, as you could see here, this green fiber, within that, the ROI is not really efficient for our study. So we propose to use another way to do it, which we call fiber-based analysis. It really is the same thing as quantitative stratigraphy, which means we segment the brain image into curvilinear regions, which singly denotes a fiber. And then, which means the voxels in these point clouds would be a sample points on the fiber, specific to fiber pretty much. So here goes our method. It's, this part should have like GUI components here, but whatever. We have 4 steps, and also we develop a pipeline GUI for neuroscientists to conveniently use our analysis framework to conduct their study. As I mentioned before, the T1, T2 raw image is not sufficient in quantitative studies, so we need to do some pre-processing of the image. The basic step follows the previous studies on T1, T2 ratio maps. We first acquired the raw data, which is commonly acquired in regular MRI. And then the 2 types of image need to be registered into the same space so that we could compute their ratio. computing the ratio itself eliminates some of the bias, which is consistent across T1s and T2. However, it's not bias-free, therefore, which makes it qualitative but not quantitative. So we need an actual step of intensity calibration, which I won't go into too much details right here, but the punchline is intensity calibration clamps the value within a certain range and eliminate the rest of the field bias that's left in the raw data so that we could use it for quantitative analysis. Here's sort of an image that shows how this process goes. The rightmost one is the calibrated image. And then the second step here is about registering these values into fiber tracts. Innovation here we did is that instead of performing quantitative cartography ourselves, we want to take advantage of the fact that there are previous DTI studies that have already performed these steps. So we got the data pretty much, which is stored in DTI Atlas with computed deformation fields. However, what we need to do is now we have T1, T2 ratio map, and we have their corresponding DTI map. We need to solve the correspondence problem, right? Which means we need to find the deformation fields from the T1, T2 ratio map to individual DTI, and then combine with the previously computed deformation fields from DTI to the global atlas space. We could efficiently register these ratio values in fiber tracts. And the way we do it is by symmetric diffeomorphic registration using the toolkit normalization tool. So here's kind of a results after this step. As you can see, this is really a comparison between the DTI FA distribution in the cortical field left parietal tract and the T1, T2 ratio distribution. And it kind of itself proves that, as you can see here in this area, certain myelination that's hidden in the FA distribution got lighted up here, which shows that T1, T2 actually reveals more information about myelination. So before we could go into the final step of statistical analysis, we need to extract a parameter profile from this fiber image, right? So the way we do it is to find a plane of origin. And as you can see here is the intersection that's crossing the fiber tracts. We take them as the 0 points, and then parameterize the fiber tracts by their arc length, so that each arc length point is associated with a T1, T2 ratio values. Generally, parameter files could be optionally used to reject some outliers. This is one case that I have rejected some outliers, and the y-axis here is the arc length, and the y-axis here is the T1, T2 ratio maps, and this is around 40 cases. So for proof of concept, we sort of conduct these example studies that show how the result could be used in statistical analysis. We use a tool called FATS, which is specific to diffusion analysis, which is easily adaptable to T1, T2. FATS provides both global and local analysis of profiles, which pretty much give you a bivariate coefficient models used in regression analysis. And we take 73 newborn subjects from UC Irvine. These newborn babies usually a few weeks old when they're at scan. And we want to ask the question, say, how age and how gender affect this myelination. And notice here that we have, by speaking of age, I actually have two kinds of age. One is postnatal age, which is usually what we know as the age of human, but measured by weeks. And also there is also a gestational age, which is the time span that the baby was in the mother's womb before the baby was born. So here's the result. Let me explain about what the plot means. First, the left-hand side of the plot is the beta coefficient plot, which denotes the covariate coefficients. And the dotted line here, as you can see, means that this part of the values at this arc length is statistically coefficient by passing the p-value test. And as we have hypothesized, premortal bundle usually have a high correlation to myelination by age. And the result shows indeed the case. that the T1 weeks, which is the postnatal age, shows significant almost across the entire bundle except some points here, but we didn't find any meaningful results from the gestational age. And I also run this test on several other fiber tracts and found some even more interesting results, which is this IFOF fiber tracts that show no correlation with other factors, but it shows a correlation with gender at this narrow band of arc-length. And also some other fiber tract that does not correlate to any of the factors we try to examine at all. So finally, I want to talk a bit about the software development part of the project. I mainly worked on 3 components of this framework. First is the core pipeline that's for use by neuroscientists, which provides a very convenient way, as you can see here is a snapshot, for you to run the entire pipeline by just clicking a few cutesy buttons. And GUI finally generates Python scripts that could be rerun anywhere to run the pipeline, publicly available on GitHub. And also 2 external modules that the GUI use. The fiber process is for transformation from ratio map to the atlas space. The tetrastat is what I have talked before, the profile extraction part. And I just want to give credit to other tools I have used, keyword ITK, VTK for segmentations and registration, shape population viewer for visualization, and FASTER for statistical analysis. All right, thank you.